Hello students, let us continue studying about the animal tissues. We have finished studying the connective tissue and the epithelial tissue. Now we are going to study about two more tissues. One is the muscle tissue and the nervous tissue. Muscle tissue is a soft tissue that makes up most of the tissues in the human muscular system. The muscle tissue is characterized by properties that allow movement. They are also responding to stimulus. When attached to two bones, contraction of muscles causes the bones to move. Attached to the bones of the skeletal system are about 700 named muscles that make up roughly half of the person's body weight. Muscle tissue is also found in the heart, digestive organs, blood vessels, etc. In these organs, muscles serve to move substances throughout the body. Muscle tissue consists of fibers of muscle cells connected together in sheets and fibers. Together, these sheets and fibers are known as muscles. Muscle tissue functions as a single unit and is often connected to nerve bundles. At the cellular level, each muscle cell has a complex of proteins containing actin and myosin. These proteins help in contraction and relaxation, which leads to movement. During winter, the body shivers. Do you know why? When the body is exposed to cold air, we shiver. So, this is because of the contraction and relaxation producing large amounts of heat. This is to keep the body hot. So, this is how vigorous contraction and relaxations of the muscles is helping us to stay warm by shivering. Functionally, we can see that muscles are of two types. Muscles that are under conscious control are called voluntary muscles. The bicep muscles, tricep muscles, quadriceps, diaphragm, etc. These are under voluntary control and they are called voluntary muscles. Muscles that are not under conscious control are called the involuntary muscles. Example, the cardiac muscle and the smooth muscles lining intestinal tract, urogenital tract, etc. Morphologically, muscles are of different types. The muscle which shows cross striations are called striated muscles or striped muscles. They are mostly the skeletal muscles. They are also called voluntary muscles. The muscles which show no cross striations are called the non-striated muscles. They look smooth and hence are called smooth muscles. These smooth muscles are not under the control of will, so they are involuntary muscles. So, on the basis of function and morphology, we have striated voluntary skeletal muscles and then we have unstriated involuntary smooth muscles. So, based on their structure, location and function, we can again classify muscles into three types. One is the skeletal muscle, second one is the cardiac muscle and third one is the smooth muscle. Let us understand the skeletal muscle. These are cylindrical, multinucleated, there are many nuclei, striated and under voluntary control. This type of muscle is attached to bone and is responsible for skeletal movements. The main functions of the skeletal muscle are movement, heat production, posture, ability to respond to stimulus, conductivity, that is the ability to transmit impulses, then extensibility, that is the ability to stretch and again come back to original shape. Then it also has the ability to shorten, that is contractibility. These are the functions of skeletal muscles. Then next is the smooth muscles. Smooth muscle tissue has no striations. These type of muscles are found in the walls of internal organs such as blood vessels, gastrointestinal tract, bladder, uterus. It is under the control of the autonomic nervous system. Smooth muscles cannot be controlled consciously and thus act involuntarily. The non-striated smooth muscle cell is spindle shaped and has one central nucleus. The smooth muscle contracts slowly and rhythmically. The next one is the cardiac muscle. This type of muscle is found in the walls of the heart. It is under the control of the autonomous nervous system. This muscle cell has one central nucleus. It is striated, but it is under involuntary control. The muscle cell here is rectangular in shape. The contractions are involuntary, strong, and rhythmical. The next type of animal tissue that we will study is the nervous tissue. This is also called the neural tissue. It is the main tissue component of the nervous system. The nervous system regulates and controls bodily functions and activities. It consists of two parts: the central nervous system comprising of the brain and spinal cord, and the peripheral nervous system comprising of the branching peripheral nerves. Nervous tissue contains two types of cells, the neurons and neuroglia, which is also called the glial cells. The glial cells support the nerve cells. Nerve cells are the only cells in the body which do not have the ability of regeneration. They are specialized cells. Cells of the nervous system are specialized for transmitting stimulus from one place to another. So there are different parts of the nerve cell. The nerve cell is also called neuron. The parts of the nerve cell are the cell body, also called the cyton, and another major part is the axon. And then there are dendrites. The cell body has a large nucleus and it has a cytoplasm within. The cytoplasm contains granular structures called Nissel's granules. The projections arising from the cell body are called the dendrites. They are sharp, branched, and they are more in number. One projection of the cyton is long, and this is called the axon. Some nerve cells, they have the axon covered with a sheath-like structure. The sheath is called myelin sheath, and there are nodes at regular intervals on the myelin sheath. The nodes are called nodes of Ranvier. Some are non-myelinated. Some nerve cells are non-myelinated. The axon of nerve cell is connected with the dendrites of another nerve cell to form synapse. This is how communication is passed from one nerve cell to another nerve cell. There are some lab activities in chapter 3 which will help us understand animal tissues better. Let us look at them. The first one is the identification of tissue in the collected sample. For this, we will require a microscope, slide, dilute HCl, forceps, and a brush. For the procedure, we will be collecting a small piece of chicken with the bone from a nearby chicken center. So the procedure to be followed here is to put the chicken piece in dilute HCl for 2 hours. After that, the skin part of the chicken piece is removed with forceps onto the slide. Another slide is kept on it and both the slides are pressed gently. So this can be observed under the microscope. What can we observe here? We can see that all the cells are similar. They are arranged close to each other and they are tightly packed. They form a continuous sheath and there is no intercellular space. So this is actually the epithelial tissue which is found in the skin. It is the covering that protects the inside of the animal body. This is procedure 1 from your textbook on page number 25. Next, we will go on to procedure 3 on page number 26 because it is again related to the chicken piece. In procedure 3, a piece of muscle of the chicken is taken and it is kept in dilute HCl or vinegar and left it for 2 hours. The muscle piece is collected next morning and it is kept on a slide. Few drops of water are placed and there is a cover slip placed on it, and this can be observed under the microscope. Under the microscope, we can see cells which are striated and with many nuclei. Skeletal muscle cells are arranged in bundles and they are cylindrical in shape. Cells of the skin, as observed in procedure 1, they are appearing flat. Then in procedure 4 on page number 27, there is an experiment to observe the bone tissue. The chicken bone can be kept in vinegar or dilute HCl overnight. Then the bone will become soft and a piece of it can be taken by using a knife. This can be observed under the microscope. The bone can be seen as something porous and with various chambers. Since this is a large surface that has to be observed, the kind of microscope used will be a stereo microscope or a dissecting microscope. Then let us come back to procedure 2 on page number 26. This experiment is to observe the blood connective tissue. For this, we have to take a sterilized needle and collect one drop of blood from a fingertip by pricking with the syringe needle. This drop is allowed to fall on a slide. Keep the finger on the slide and collect one drop of blood on the slide. Another slide is placed on it gently and this smear is observed under the microscope. So in this smear we can observe various blood cells. Here we can identify mostly red blood cells. All other cells will be there, but only the red blood cells may be visible. To observe other cells in the blood, we will need different procedures. Various diluting fluids are required. So for this procedure, in this procedure we can observe only the red blood cells. The next activity, which is there on page number 27, is to collect the substance lining the mouth using a spoon. And this lining can be observed under the microscope. So when we scrape the inside of the mouth, what we get is the oral mucosa. The oral mucosa is made up of stratified squamous epithelium. The cells are arranged regularly.